Brilliant. Thank you so much. Welcome, everyone. My name's Nick Fuggle. I'm a clinical academic down at the University of Southampton, for anyone that I haven't met. Welcome to this latest edition of the Clinical AI Interest Group, having had a brief hiatus over the summer. I'm really excited that we're doing a joint meeting with the Omics Interest Group, also from the Turing, and we're joined by Owen Rackham, who's going to speak to us today. For the uninitiated, the Clinical AI Interest Group is for anyone who's interested in the deployment of clinical artificial intelligence, and is based around three main themes. One, pooling, being a pool of resources that can feed into other Alan Turing Institute activities. Two, sharing, so being a forum in which we can share developments within the field and things that we're interested in. And three, education. And so if AI is going to be truly deployed in clinical practice, then we need to make sure that health professionals understand the tools that they're using, not just well enough for their own comprehension, but so that they can explain it to patients so they can be truly consented. So that's the Clinical AI Interest Group. Owen, do you want to do a little introduction to the Omics Interest Group?
Sure, yeah. Very similar sort of setup. We're a group of people whose common thread is an interest in some way, shape, or form in either the creation or the use of anything omics. So that's basically proteomics, single cell sequencing, images, anything that's sort of basically large data sets collected about biological systems. We typically have 3 in-person meetings a year, and then some online meetings where we sort of discuss a range of topics, right the way from like current research or like experimental design, things like this. So yeah, it changes and varies, but all related around how to get the most out of the rather large amount of money that people are spending on creating omics data, basically.
And I can actually speak from experience because I am also a member of the Omics Interest Group, and have been to a couple of the meetings, and they are really brilliant. So definitely to be recommended, and a lot of clinical overlap as well. We're going to have some breakout rooms in a moment as an opportunity to network. But before we do, just a few brief updates and notices. Firstly, if you're joining us for the first time, we have the interest group, but we've also got supra interest groups. So if you have a particular interest in a different clinical field, then please do engage with those. At the moment, we've got those in computer vision and medical imaging, in pathology, clinical pathology, in ITU and anesthetics, and in women's health. And they're actually really dynamic, and lots of events are going to be springing up. And in fact, in our November edition, that's going to be hosted by the Computer Vision and Medical Imaging Super Interest Group, and who'll be hosting a debate or a fireside. That's right, a fireside, isn't that right, Emma? Brilliant. So that's the Super Interest Groups. And then we held our first summer school, which was attended by, I think, about 30 attendees. We had to move from in-person to virtual because of train strikes, and also because of junior doctors' strikes at the same time. But we've got a second part of that coming up, in which people actually get to play with Python code and to develop their own clinical artificial intelligence tool. So exciting things going on there. Anyway, Joe, I'm just going to hand over to you to introduce the introduction section.
Thanks very much, Nick. And apologies in advance if I sound a little bit more gravelly than normal. I'm struggling with some sort of lurgy, which, despite recent history, apparently isn't COVID. Something else, and something worse somehow. So we're going to spend the next 10 minutes or so in breakout rooms. We're going to go into, I think, about 5 or so per group, and this is an opportunity to network as a small community. So to get to know a little bit about each other. And to give this next section a little bit of structure, we wanted the small groups to consider the following questions. So the first one is, who are you? Second one is, where are you from? Third one is, how does AI fit into your work? And then finally, if you have any asks for the wider group, so anything that we can help with, anyone on the call, and also if you're looking to recruit or for any collaborators, this is also a great opportunity to ask that now. So I'll post those questions into the chat so everyone has those, and in the next few moments we'll all join our breakout room. So thanks everyone. Hope that was enjoyable. Obviously, if there are any conversations that are ongoing and would be useful to pick up offline, feel free to either chat in the background on this call or even to open conversation on our Slack channel. And for anyone that's not managed to join Slack yet, we'll get some details across. But it's a great place to connect and have these conversations. So it's my great pleasure now to introduce Owen Rackham, who's our speaker for today. So Dr. Rackham is an associate professor of systems biology at University of Southampton. And he leads an interdisciplinary team working on combining artificial intelligence with high throughput biology. So that's, as Owen already mentioned, the kind of the multi-omics techniques to better understand health and disease. And he's also the lead or co-lead for the Turing's Omics theme. So Owen, thank you so much for joining us today and for finding time to speak to us. And I'm going to hand over to you to introduce your talk.
Thanks, Joe, and thanks for inviting me to come along and speak. I have to say, I'm a big fan of the interest groups set up at the Turing. It's a really great way to meet people doing different things. So I'm very glad to be here. Let me just try and share my screen, and then hopefully should have been. Find the right talk. There we go. So hopefully you see my slides. I'll just check if that's okay. If someone could just let me know.
Perfect.
Yes, we can see your slides.
Perfect. Okay. And I should apologize for my 1980s aspect ratio. So I don't know why they look like that, but that's how it is. So yeah, I'm based at the University of Southampton. I also have this secondment into the Turing Institute, where I'm the theme lead for cell and molecular medicine. What I wanted to do today, because we're a very broad group of people, but I guess very broadly brought together by our interest in AI, is actually describe a couple of pieces of work that I've done, but use that as a way of sort of motivating where there's a sort of gap in the capability for AI and data science at the moment. So because of that, I'm going to try and explain a lot of things quite quickly. So I won't give a lot of technical content, but I'm happy to talk about that as we go through. I just want to give a feeling for what I've been doing and motivate the sort of challenge that I'll talk about at the end. Okay, so the place to start, I guess, is that we tend to think of the cells in our body as being rather individual, rather separate. We think of them as having specific functions, having specific gene expression profiles, containing certain proteins. And the sort of reason for that is that we have sort of observed them doing things over time. But I think what we're sort of starting to understand is that actually the flexibility in cell type is actually rather high. The cell types that we have, we do seem to be able to control them to some extent. And I'll just demonstrate that by showing you an example. Sorry. So I'm using this analogy that actually what I think is that all cell types exist somehow on a continuum, and that's what I'm using this landscape to represent. So each position on this landscape would be a different cell type. So for instance, in this particular position, there may be a fibroblast, and if you take that fibroblast and you culture it, this is what it looks like. But I think what's fascinating is that if I take those fibroblasts and I overexpress 3 transcription factors only, what happens is that fibroblast will change its state. The gene expression profile of that transcription of that cell will change. And actually what will happen is it will transform itself into something that looks an awful lot like a cardiomyocyte. So if I now look at those same cells down the microscope, what happens is that they've really changed. Their phenotype is completely transformed. They were fibroblasts, and now they are beating cardiomyocytes in the dish. And I think this is what a really clear example of the flexibility of cell types. And actually the flexibility that is possible with actually a relatively small number of perturbations. So in this case, just 3 transcription factors. It's not only fibroblasts to cardiomyocytes. Very famously, Shinya Yamanaka, he actually won the Nobel Prize for discovering that these 4 other transcription factors, if I put them into a fibroblast, what happens is that fibroblast will become an embryonic stem cell-like state. This is this induced pluripotent stem cell that you may have heard of. So this is obviously a very powerful phenomenon, the fact that we can control cell type in this way. And I just sort of mentioned a couple of those very quickly, like we're starting to see more and more cell-based therapies come into the clinic. And actually, a lot of those, or the ones that, certainly the ones that were in the clinic first, were cell types that were extracted from patients, maybe just put straight back, or maybe in some way they were edited in order to take on a new function and then put back in. So maybe you've come across things like CAR-T or CAR-NK, or just even like a HSC cell therapy. But knowing that we can control cell fate in this way means that actually we have the capacity potentially to make any cell type and use that as the basis of a cell-based therapy. And also something else that we're starting to increasingly see is that we've had gene therapies, but we're starting to see the development of a new class of gene therapies, which is essentially an in vivo reprogramming. So in many diseases, but I'm just using an example of the eye here, some injury or some genetic abnormality might mean that you get a selective loss of a certain cell type. So maybe you lose the photoreceptor cells in your eye, and that obviously leads to blindness. We're starting to see the development now of, excuse me, pressing the keys too much quickly, of the development of gene therapies, where the gene therapy is actually the delivery of transcription factors which induce the conversion of other cell types into the cell types that are lost. So this is sort of an in vivo reprogramming approach to regenerative medicine. So I think there's a huge potential here. Like, we now understand that we can control cell type, but there is this problem that still exists, which is that there are well over 300 cell types. Every week, a new paper comes out claiming to have discovered another one. And it takes an awful, there are about 2,000 different transcription factors. So being able to manipulate cells systematically, and by that I mean knowing how to go from any place on this landscape to any other place on this landscape, is a huge combinatorial exercise. And it's not one that we can do just by trial and error. This is a data science and an AI challenge, essentially. And I'll show you some work very quickly, which we have done to try and move towards that. But it's by no means a solved problem, if you like. So the work I want to tell you about very quickly is trying to answer this question of how can we systematically work out which perturbations to use to make any cell type. And the solution we have to that is this algorithm is called Mogrify. And I'm just going to very briefly describe the sort of theory behind Mogrify rather than the sort of technical details of how it works. But just so you get an understanding of the sort of possibilities that exist from using a sort of data science AI approach to these problems in biology. So clearly, every cell type in our body has a different gene expression profile. So the first thing that we did as part of this work, and actually was part of this consortium, the Phantom Consortium, we collected gene expression profiles of about 1,000 different cell types. This is in an era before single cell sequencing, I should say. And then we're going to take just that data, but we're going to apply an algorithm that basically encodes 3 steps of logic in order to predict from those different gene expression profiles what I have to do to one cell type, as in what transcription factors should I add to one cell type, such that it will convert into another cell type. Okay, so the first step of that is that we want to know for every gene, how specific each, for every cell, how specific each gene is. So clearly there are some genes that are very, very cell type specific. They confer functions which are only useful in one condition. So neurons, for instance, express very particular receptors on their surface which only neurons use. And equally, there are some genes which every cell type is using, like ribosomal proteins, for instance. So the first thing is that across every cell type that we had, which was a fairly decent representation of the cell types in human, we first calculate a score, which basically says for every gene, how specific is the expression to a particular cell type. Okay, so the second thing is that what we're trying to do here is identify regulators that we can introduce to one cell type that will induce a change in expression. So I guess you can think of this a little bit like trying to start a revolution, right? We're trying to move the gene expression profile from one state to another. And what we already knew is that just by looking at the specificity, it's not possible to identify the factors that we know can induce the cell fate change from one cell type to another. So what we asked the algorithm to do was instead look for regulators that are not only specific, but also they appear to be very highly connected to other genes that are also specific. So that means in my analogy of starting a revolution, we don't pick the person who has the most polar opinion. Instead, we try to pick the people who have the stance that you're trying to create, but also we know that they have a lot of influence. So basically, we're trying to find transcription factors in this case, where we're very sure that if we overexpress those in a starting cell type, they will influence a lot of the genes that need to get turned on or need to get turned off in order to go from one cell type to the other. So then the third thing that this algorithm does is, because of the way that nature has evolved, there is an awful lot of redundancy in the regulation of gene expression in our bodies. And that means that if I do step 2, and I say, for instance, I want to find the transcription factors to make cardiomyocytes, it's very likely that many of the top ranking transcription factors all regulate the same genes. But if we're just going to pick a small set of transcription factors to overexpress, we would much prefer to pick ones that don't regulate the same thing. We would prefer that their coverage of the genes that need to change in order to go from a fibroblast to cardiomyocyte are different. So the algorithm also has this idea of trying to reduce redundancy in terms of the transcription factors that we select. So I'm sorry, that's a very, very brief description of the algorithm, but I guess for the purposes of today's talk, what you can think of this as being is it's an algorithm that if you can sequence 2 or more distinct cell types, then it's an algorithm that can be applied to predict what are the transcriptional regulators that can drive a conversion between those cell types. Okay, so that's the sort of background. That's the hypothesis of this algorithm. The idea that it's designed to encode, but obviously it's only really useful if it works. So it's been tested on several things. I just sort of show you a few of those. So the very first experiment that we did actually is that we asked Mogrify to predict how to convert a dermal fibroblast into a keratinocyte. So this is a conversion that's never been achieved before. We haven't been able to find by experimental trial and error those factors. So the algorithm makes its prediction. What we do actually is we take that prediction, which is a set of transcription factors, we put them into a virus, and then we infect the source cell with that virus in order to overexpress the transcription factor. So in this case, what you're seeing on the top row are cells that were keratinocytes that have had those predictions overexpressed, and they take on this morphology of a keratinocyte, which we call this cobblestone morphology. And obviously, we check that they have the gene expression profile. They behave in every way that we expect a keratinocyte to behave. They're starting from a fibroblast. Equally, we said, can we start in a keratinocyte and instead create a microvascular endothelial cell? These are some of the cell types that make up the vasculature in our bodies. And again, we asked the algorithm to predict what are the transcription factors. We put them into a virus, we overexpress them in keratinocytes. I just put one microscope image as an example, but those cells take on the behavior and the phenotype and the gene expression profile of endothelial cells. So it seems like at least between distinct cell types that this algorithm is capable of predicting transcriptional regulators that can induce change between them. And just to say that one thing that we realized having developed this algorithm is that as far as the algorithm is concerned, a cell type is just anything that is stable enough that I can assay the gene expression profile. So having used it to convert between cell types of different states, so between like fibroblasts, cardiomyocytes, for instance, we've also used it for a number of other things like converting cells from an aged to a young state in mice. We did that in intestinal stem cells. We used the same approach to look at young and old cells and work out what are the transcriptional regulators that would move you from a young, from an aged intestinal stem cell to a young intestinal stem cell. We've also used it for predicting how to induce differentiation in cancer states. Presumably many of you know, but one of the hallmarks of many cancers is that actually what happens is that the cells get stuck in an undifferentiated state, and that means that they proliferate. So one option for treatment of cancer is that you work out how you could induce those cells to differentiate so they stop proliferating. And we've also used a similar approach to, rather than look for transcription factors, to look for drugs that can do the same thing. Okay, so I just reveal this slide. I'm building towards like capabilities that I think we have, because I want to try and motivate a challenge that I think that we have as an omics community still outlying. So I guess what I've shown you is that cells are super complex. They do respond to many things, many millions of different things actually, including their environment. I've shown you that for transcription factors, at least, we seem to be able to make some predictions that will allow us to systematically convert between cell types. But we're still a long way from being able to do this for everything, for all of the different things that cells respond to. And being able to do that would be a fantastically important model to produce. If we were able to predict what the transcriptional response to any drug was, or what the transcriptional response to any protein was, this would be a fantastic way for developing not only a better understanding of how diseases develop, understanding how changes in our environment might induce the onset of disease, and also for looking for novel treatments to disease. So that's sort of, I guess, one thing that I want to point out, and I'll come back to why I think that's a challenge later on. But I just want to also use another piece of work that we have done in collaboration with a group in Australia, actually, to demonstrate another phenomenon of cells that I think we also really well understand, but that we don't have a particularly good way of systematically controlling. So I showed you already this, we have this idea that all cell types exist on a landscape. And I showed you the experiment of going from fibroblasts to cardiomyocytes. I mentioned already that Shinya Yamanaka won the Nobel Prize for discovering 4 transcription factors that will convert fibroblasts back to the state of pluripotency. But for a long time, we didn't really understand like how that was happening. We knew that overexpressing those 4 transcription factors was sufficient to convert the fibroblasts back to pluripotency, but the steps involved in that, like what molecularly was actually going on after those transcription factors had been overexpressed, we didn't really understand. So we set out to better understand that. And actually, like many interesting scientific discoveries, we actually found something entirely different. And it's something entirely different that I want to share with you as a demonstration of how flexible cells are, and how exciting it would be if we were able to have a better understanding of how to control that. So what we did is single cell sequencing came along. If you're not familiar with this technology, basically it's a way to assess the abundance of RNA, so how much of every gene is being expressed in each individual cell in an experiment. So in this case, what we had is we had fibroblasts, we overexpress the 4 transcription factors that we know induce this conversion back to pluripotency. And then we use single cell sequencing to basically trace the progress of cells going from the bottom left in this reduced dimension plot of this process, either the ones that don't reprogram, this cells that we call refractory, or cells that actually they reach 2 different states of pluripotency, and I'll explain the difference between those in a moment. But basically, we use single cell sequencing to understand. We were trying to understand what the process was, what was happening when we overexpressed those transcription factors. But because of what we found, I just have to do a very, very, very brief developmental biology 101. So forgive me for the brevity of this, but I need to just explain a couple of things in order to highlight what we found out. So pretty much the earliest thing that happens in human development is we start from one cell, that cell continuously divides, but it's becoming specialized over time. And actually, the first thing that happens is we get some cells that become the epiblast, and those are the cells that will go on to create the embryo, and some cells that become the trophectoderm, and those are the cells that go on actually to create the placenta. And when we talk about embryonic stem cells, we're talking about the cells of the epiblast, because they are the cells that will differentiate into each of the cell types in our body. They're the ones, the somatic cell types that we have. Once that blastocyst forms, one of the first things that it does is it implants itself into the uterus. So we think of the blastocyst as having a pre and a post implantation state. So these 2 states that I was just showing you here, these naive and prime states, they represent cells that mimic the pre and the post implantation state. But they both, importantly, are states that come after the trophectoderm, which is this first decision that gets made by our cells, whether they will become part of the body, part of the embryo, or part of the placenta. So what we expected when we would do this experiment is that we would observe cells going to one of these 2 states, either to the pre or the post implantation like epiblast cells. But to our surprise, amazement, I'm summarizing about 3 years of work in 10 seconds, but what we realized is that actually also during reprogramming, there is this very small population of cells that look like they're also becoming the trophectoderm. So that means that during reprogramming, as in once we overexpress these 4 transcription factors, some cells don't reprogram, some cells become the pre-implantation epiblast, some become the post-implantation epiblast, and some cells become the trophectoderm-like cells. So just to convince you that that's the case, we're able to take those cells and culture them in conditions that we know support trophectoderm. Well, actually, in this case, trophoblast stem cells, the progenitors of the placenta. They look and behave like trophoblast stem cells. They differentiate into all of the cells that we expect to see in the center. And one very nice way to also test this is actually if you take a pregnancy test, what you're doing is actually testing for the presence of a placenta, not for the presence of an embryo. So if you take these cells that we create during reprogramming and you put them in subcutaneously into a mouse, and you take the urine of that mouse, it will make a pregnancy test positive, which is a great validation that these cells are placenta, and also made for a very interesting morning for the postdoc who had to go and buy an awful lot of pregnancy tests in one go. So already this revealed something that we were not expecting, which is that we thought we were creating cells from the epiblast, but actually we were also creating cells from the trophectoderm. And I won't talk about it in detail, but actually as it transpired, we were also creating cells from this other part of the blastocyst, which is this primitive endoderm state. So it seemed like during reprogramming, all three of these different parts of the blastocyst were being created. So in collaboration, led actually by someone from Monash University, we wondered if we could take cells that were undergoing reprogramming and put them into conditions that mimicked 3D conditions that mimicked early development to see what would happen. And actually what happens, and again, I'm condensing an awful lot of work into a very short period of time, is if you put those cells into a 3-dimension conditions, they self-organize themselves into something that looks a lot like the blastocyst. It's a model of the early blastocyst, which we named blastoids in this case, or induced blastoids. And again, I won't go into too much detail, but they look like a rather good model of a blastocyst. So this is an organoid, if you like, of early development. They contain all of the different aspects that we know an early blastocyst should have, and they respond in ways that we expect blastocysts to respond to, if you like. Okay, so forgive again the brevity of the description of that work, but what I wanted to use it to demonstrate is that not only are cells complex in the way that they can respond to perturbations, and that's a hugely combinatorial problem involving many millions of different ways of perturbing cells, but also cells by themselves are complex, as in if you put them together in the right conditions, they will assemble themselves in such a way as to confer function. So in the work that I just showed you, if we put the right cells with the right perturbations in the right conditions, they would assemble themselves into the 3-dimensional structure of the blastocyst. So the purpose of that very brief description of all of that work is just to basically say, we know that cells are very, very plastic and that they can be controlled. And actually now we have a lot of tools for measuring that, for observing that. But we still really very much are in our infancy in terms of being able to predict the effect of perturbations. And also we know that if you take cells and you put them in the right environments, that they will organize themselves into structures that have function. And again, we're starting to have a lot of techniques for observing that. Like we've always had microscopy. Now we start to have more sophisticated tools like spatial transcriptomics, where we get to see the result of cells organizing themselves, but we really don't understand at all actually how they do that. And we really, really don't understand how to control that. We're a very, very long way from being able to engineer these 3D structures of our own creation to try and make, I guess, sort of cellular systems for treating disease, or cellular systems for modeling disease. So I hope that all makes sense. And now it sort of gets to the point where I sort of want to pose the challenge that we have as a data science and AI community, which is basically what we're talking about here is engineering. Essentially, what we should aspire to want to do is to be able to engineer cellular systems in the same way that we engineer other systems, like systems in the physical world. Our capacity to engineer bridges is much better than our capacity to engineer cells, and I think that's for lots of reasons. But I think one of the sort of fundamental things about modern engineering is that we're very often relying on models of the system in order to be able to create in silico first to make sure that we can create the things that we want. So for instance, if I'm engineering a bridge, I don't just observe lots of bridges and hope that having observed them, I know how to create the bridge that I want. What I do is I make a model that encodes the laws of physics, and I build a model of the bridge that I hope to create, and then I make sure that it's going to do all of the things that I want it to do. Like it's going to be able to support the weight of the traffic, it's going to be able to stand up if there's a strong wind. And it's really like this level of engineering that I think that we should aspire to be reaching in biological systems. And I really think that that is a problem for AI and data science rather than experimental biology at the moment. And I'll just try and motivate that in 1 second. But clearly, bricks are much simpler than cells. Although if you tell an engineer that, they may disagree. But like I said, cells are complex in their own right. They respond and change to their environment. But I think that's the one thing that I would like to sort of emphasize is that if you look at the sort of progress and the investment that's been made already, it makes it very clear where the gap is. So we now have an awful lot of ways for manipulating cells. We have small molecules, largely thanks to COVID, we now sort of start to see an explosion in RNA-based therapeutics. We have always had antibodies, all of these kinds of things that we know we can use to manipulate cells. We've also invested billions and billions of pounds in observing cells. So just taking one of these as an example, the short read archive is where everyone deposits data that they've generated, which basically is describing anything that comes out of a sequencing machine. And there's really tens of billions of pounds of data in there. But what we don't have is a very good way for connecting these things. We don't have very good ways of taking this large investment in observing cellular systems and using it to work out how to deploy these methods we have for manipulating cells. So I guess, like I said, I wanted to end on a challenge. I think I've taken far too much time, but I'm sorry about that. And the challenge is sort of this, like at the moment, there's this huge gap where there are communities like those who are interested in engineering cellular systems for creating cell therapies or building models of disease. And there is all of this data over here, literally hundreds of petabytes of data. But the people on the right-hand side are not able to interact with all of this data. They're not able to benefit from it. So I think that there is this, we have this challenge at the moment about how to close this gap. And I think that really what we should aspire to be doing is that people in this community, these people that are engineering cells, they shouldn't be interacting with this data directly. They should be interacting with models that are trained on this data that allow them to ask questions that are useful to them. They ought to be able to ask questions like, what is the effect of knocking out gene X in cell type Y? And I think that there are lots of things that we need to do, but basically they fall into these three categories. One is, how do we wrangle this data and integrate it? One is, how do we build foundation models that are suitable for biomedicine? And then how do we train people both to create those, but also then to utilize them? And I'm conscious I've gone for too long. I didn't do that all myself. Here are just some of the people that did the work, but I want to stop so that we have time for some questions, if I can find the stop sharing screen button. There. Thank you.
Brilliant. Thank you so much, Owen. There was a lot there, but you went through it really in such a kind of tangible and understandable way that even I was able to keep up with most of it. If anyone's got any questions, please do pop your hands up. But I had a few. One was particularly with regard to like the blastoid. So you talked about how you end up at the blastoid cell, but where does it go? Where does it go from there? Does it just stay in that state, or does it continue to develop further?
Yes, well, you won't be surprised to hear that the creation of those things is extremely heavily regulated. So actually, you cannot culture them for longer than, there's a very defined cutoff point after which you can't culture them. And actually, this work was done in Australia, and in Australia, cloning is against the law. So actually, a lot of regulations had to be created in order to allow the future development of this work. In the UK, that's a little bit different. Because they are derived from skin, we actually don't consider them like embryos, so they're regulated slightly differently.
Mhm.
But you are allowed to do certain things, like you can run an implantation assay, for instance, so you can model implantation in vitro and see if they respond to that as you might expect. But you're really limited in terms of the days that you can do anything with those. But I think the sort of exciting thing about them is that clearly getting blastocysts is fraught with ethical and operational problems, right? Actually, really the only way to do that at the moment is from IVF blastocysts that aren't used in IVF. So here we maybe have the opportunity to create at scale a model of the blastocyst. So you really like hundreds or thousands of these things. And that means that we can start to test like what are the effects of environment on early development? What are the effects of genetic abnormalities on early development? So yeah, but it's really, this work is coming out in the last year. So everyone's sort of still trying to work out how to regulate it well and how to use it in the most powerful way.
Oh, that's very important. Joe.
Yeah, thanks so much, Owen. I had a couple of questions, a little bit philosophical, so apologies in advance for that. Without asking you to predict the future, which I know is impossible and fraught, what do you think are the key kind of disease areas or clinical problems that taking a kind of cellular engineering approach will help with in the near future?
I mean, I'm heavily biased, right? Because I started a cell therapy company. So clearly my stance on that is that actually all diseases are essentially progressions of cell fate change, right? As in we at some point we're healthy and then we're not. So actually I think that you should be able to use these kinds of ideas to in all of the different indications. I think that some things are more obvious than others to happen sooner rather than later. So for instance, in the eye, like the loss of cells in the eye that lead to blindness, I think that that's probably a relatively close ambition, right? As in replacing those. And we're already seeing people doing things like making beta cells and then introducing beta cells as a way to control, to control diabetes. But I think that that's because we're seeing those first because they are the most obvious, and the sort of route to treatment is also quite well defined. But I think one thing that we've seen, for instance, is that we can reverse aging. We did it in mice, but you can reverse the age status of an intestinal stem cell. So there's no reason that we shouldn't be able to reverse other things that develop over long periods like that. But I'm biased. You should ask someone else that question as well.
Thanks. I had a quick follow-up, if that's all right. Do you see cellular engineers as being future healthcare professionals, or are they going to be almost like drug developers? What do you think? Are they going to be regulated in that same way, or providing a clinical product?
So I think that cell engineers, maybe not clinical, but I think that we should be aspiring to be able to understand at the molecular level things that we are seeing in the clinic, right? Like the way that we treat people at the moment is in very large groups of people that are basically grouped together with quite crude biomarkers often, right? I think that as our capacity to collect data, and here I'm talking quite a long way in the future, right? I think it's not impossible to imagine the situation where we're using these ideas of cell engineering to help us better understand at the individual level, like why diseases progress and what the best intervention might be. So I don't think they're ever going to be like clinical as in holding clinics, but I do think that these ideas will get closer to the clinic in terms of helping people understand why groups of people or why individuals develop disease the way that they do and how best to intervene.
Brilliant. Owen, thank you so much for joining us and for sharing these insights. We're going to draw things to a close now. So just to say that the next Clinical AI meeting is on Tuesday, the 10th of October at 12 p.m. And that's going to be with Mark Sendak, who's from the Duke Institute for Health Innovation. They've got this really interesting setup there where the university and data scientists have access to some of the data that comes live from the hospital, and they can develop algorithms and then test it directly within there. So it's a really interesting model, and one that, you know, we're looking to try and recreate in certain ways in the UK. That's the clinical AI point of view. Owen, I know that there's an exciting Omics event that's on the horizon as well.
There is. It's actually about advancing virus livestock omics. So I don't know. It's a bit of a departure from the clinical space, but it's really exploring how, like, tools from omics, which largely actually are developed in mammalian systems, human systems, how they're being applied to understanding virus interactions in livestock, for instance. Is on the 28th of September. But happily share. Please keep an eye out on other omics groups that might be more closely aligned with the clinical AI in the future. We can point to those.
That'd be brilliant. Right. Well, just leaves me to thank Owen once again. Thank the Omics interest group for joining us, and look forward to seeing you next month.